Hey guys, it's Mr. Wolfe here, and I'm going to be going over the process of meiosis. Not only that, but comparing it back to mitosis. So before we go into this new process of meiosis, let's review on mitosis in terms of what its purpose is. So for mitosis, the purpose is for growth, repair, and if you are a single-celled organism, asexual reproduction. This is how you're going to reproduce. So let's add a little arrow that should already be there. All right, so with mitosis, we're going to start off with a diploid cell. So I'm going to start to draw my chromosome. So hopefully you guys have two different colored writing utensils, whether they're pencils, pen, doesn't matter. In this case, I'm doing red and blue. And so in my first cell, I'm going to draw a small red chromosome and a large red chromosome. And then I'm also going to draw a small blue one and a large blue one as well. Okay, and what are these chromosomes called? Well, these are going to be called homologous chromosomes. So I'm actually going to color this one in right over here and blue for the other one. And if we had to give a quick definition for these guys, we would say that homologous chromosomes are a pair of chromosomes that share the same genes, but they may have different versions of each gene. So why do we have a pair of chromosomes? Well, we get one from the mom, and we also get one from dad as well. And what do I mean by they share the same genes? Well, let's say that they maybe both of these, I'm going to maybe draw this like black line right here and a black line right here. Maybe that's the part, that's the gene on that section of the chromosome where we have the gene for eye color. All right, so same gene, but maybe the dad has the gene for blue eyes and the mom has the gene for brown eyes. Okay. And now we have like a little bit of review on the homologous chromosomes. Let's go back to mitosis. All right, so with mitosis, we're going to start off with a diploid cell, and a diploid cell is just going to be a cell with two sets of chromosomes. All right. And so what do I mean by that here? Does it mean that I have like two sets here? No, it means I have 1, 2 small chromosomes and 1, 2 big chromosomes in this case. So I could label this cell, this diploid cell, since it has two sets of chromosomes, my N is a set, it has two sets, so it's a 2N cell. And the first thing that's going to happen in mitosis is DNA replication. And notice how I am kind of having it overlap into meiosis as well. So DNA replication is going to happen, so I'm going to have all my chromosomes replicate. And now I have, let's see, 1, 2, 3, 4 small ones, 1, 2, 3, 4 big ones. So I have 4 sets. This is technically a 4N cell. We would call it quadruploid. All right, so in mitosis, let's talk about our goal is to make my result is going to be 2 genetically identical diploid cells. And we're able to accomplish that because we replicated the DNA, so when the cell splits, each cell gets its own copy of a chromosome. And my cells at the end look completely identical, and they're also completely identical to the beginning cell that we had as well. So that's mitosis. Now let's look at the similarities and differences with meiosis, our new process. So the purpose of meiosis is to create or produce sex cells, so sperm cells or egg cells, but our scientific name for that is going to be gametes, and it's all going to be for the purpose of sexual reproduction. And before I go on with here, just since we have 2 sets of each chromosome, my ending cells are going to be diploid, so 2N over here. All right. Now for meiosis, I'm going to start off with a diploid cell, so it's going to look exactly like our beginning cell in mitosis. And not only is it going to start off like mitosis, the first step is going to be very similar to mitosis as well. Meiosis is going to do DNA replication too. However, notice how I'm drawing how my cells or my chromosomes line up here. It's important that you guys do it like this too. Okay. So the purpose of meiosis, before we go on, is to produce, I'm just going to say to produce, or I'll just say the result is 4 genetically, and in mitosis it was identical, in meiosis it's going to be 4 genetically different haploid cells. Right. So these cells being different explains why your brothers and sisters, if you have them, look similar to you, because they're going to look similar because you guys get DNA from the same source, right, from your mom and dad. But by making these cells genetically different, you're getting a different half of your mom or dad's genes, and so you guys are going to have different genes even though they're coming from the same parent right here. Okay, so how does the cell actually accomplish it? How do you actually make these cells be genetically different? Well, it happens right after DNA replication in these 2 steps right here. The first one we're going to call independent assortment. Okay. And all independent assortment is the fact that the chromosomes line up, but how they line up. So chromosomes line up independently of one another. So what does this mean? Well, I like the analogy of, think about if you're ever like in gym class back in the day, like in junior high or in summer camp, when you would make teams, the kids would line up and you would count off 1, 2, 1, 2, 1, 2, right? And you would have one team here, one team here. Well, imagine it was the next day, so maybe that was on a Monday, and on Tuesday, you come back to camp and kids line up for a new game, and you go 1, 2, 1, 2, 1, 2, 1, 2. Most likely, those 2 teams from Monday and Tuesday are going to look completely different because the students lined up, for the most part, independently of one another. And let's say we go through that entire summer, most likely you're going to always have a different team than you had before. So the same thing is going to happen in the cells. How they line up right here, right? In this case, I just happen to have them line up red and red on both sides, but the blue could have easily gone on this side as well. And so when they split, you could always get these different combinations. All right? And this actually produces 2 to the 23rd power, which I think is close to something like, I think it's 70 trillion different possibilities you could get just by lining up differently. And we also showed it in this example, right? I could have the blue be on both sides, or I could have the blue be here and the blue be here. So when they split in these different scenarios, you're going to get these genetically different cells. And so we're going to say independent assortment creates genetic variation. And I'm running out of room. So hopefully you guys did better. All right. So there's going to be another process that also creates genetic variation. So once they do line up, what we have is a process called crossing over or synapsis. And what this means is, well, let's start coloring this one in red. And I'm only going to color in this one red up to here. Because in crossing over, when they do line up, the chromosomes will actually kind of cross over, that's the name of the process, and they'll swap parts. So when they cross over, this part of the mom's chromosome is going to kind of hop over to the dad's, and the dad's part, or the dad's top of his chromosome, is going to hop over to the mom's. So again, what we're doing here is we're kind of just shuffling the genetic deck, right? We're creating all this variation, so when we get these resulting cells, they're all going to be genetically different. So if we just had to give it a quick definition of crossing over, we're going to say copied chromosomes swap parts. And I'll show that by kind of drawing this arrow right here. Okay, So now we've created a lot of this genetic variation. All we need to do now is to create our haploid cells. Because when they've replicated, I'm now at a 4N cell, right? 1, 2, 3, 4 small chromosomes, 1, 2, 3, 4 big chromosomes. All right, and the cell's going to split. And remember, since we did do this crossing over, these chromosomes, just small and large one, are going to be fully blue, but the ones over here are going to be a little bit smaller in terms of the blue because they're going to have these red tips right here. Because the chromosomes swap parts. Oh, shoot. I realize I kind of messed up right here. So I'm going to cross this out because it's the red on this side, right? Same thing, though. On the ones over here, I'm going to have the fact that the blue swapped over. And then on this side, full blue on the right. But then over here, it's a little bit shorter because the red swapped over as well. Okay. So now I have these diploid cells, right? And if we look, this is kind of like similar to the process of mitosis in terms of like the splitting and everything where we're left with these diploid cells. But if I even compare mitosis to meiosis at this point, they're different, right? Here we have identical cells, and if I just look at these cells, they are genetically different because we did this independent assortment and this crossing over. Okay, now what's going to happen is since we need these haploid cells is they're going to split again. So draw my full small and large red chromosome here, and then over here small and large red chromosome, but they're going to have a little bit of that dad chromosome that swapped over. And a little bit of the mom chromosome that swapped over here. All right. So in this case, I now have my results. If we look at these cells, none of them are the same. They are genetically different. Not only that, but they're also haploid, right? If I count how many small ones I have, 1 small chromosome. How many large chromosomes? 1, right? So this is just going to be one set of chromosomes in all of them, haploid cells. And so this is the process of meiosis.